What we're going to talk about today is just a wide variety of different microorganisms and look at certain salient characteristics of these different groups of microorganisms. So I've just taken, bits and pieces from this chapter. My version is different, from the chapter. I have a table which on your handouts, the things in red up here are underlined on your handouts, and they represent the most important things, although obviously the whole table is important, but certain things are more important than others. So we're going to talk about all these different groups. In particular today, we will focus on gram-negative rods and staph and strep, because they are all especially important medically. But our first group is the spirochetes, which we have talked about before. These are gram-negative helical flexible spirals. They are motile. Remember those axial filaments, the endoflagella. And some spirochetes are pathogenic. I've talked about these two organisms before, Treponema pallidum, which causes syphilis, and Borrelia burgdorferi, which causes Lyme disease. We also have some other spiral-shaped organisms, which are aerobic or microaerophilic. They're also gram-negative. These tend to be rigid spirals. They're also motile with endoflagella or maybe flagella that stick out. And some of these are pathogens. Campylobacter is an example of an organism that's associated with food poisoning, in particular when it comes to handling raw meat like raw chicken. That's one of the reasons why after you handle any raw meat, you should wash your hands. After you cook that meat, whatever plate or platter that you used, you should not put the cooked meat back on that same plate or platter. You should either wash off the platter or get a new one, right? You don't want to put your cooked meat back on the same platter that had the raw meat on it. And make sure you wash off everything, including all utensils, well. Now, Helicobacter pylori is an interesting organism. And can anyone tell me what part of the body is infected with Helicobacter pylori?
The stomach.
Right, the stomach. And so this is an organism associated with ulcers. So stomach ulcers, in the past, your doctors would tell you you're too stressed, you eat too many spicy foods or fatty foods, and then they would give you antacids. And in reality, what those people needed were antibiotics. So many ulcers are actually the result of a bacterial infection by Helicobacter pylori. But the interesting thing about this organism is that when they tested a bunch of different people, they found a significant percentage of humans do have Helicobacter pylori in their stomach, but they do not suffer any negative consequences. So most of those people did not get ulcers and were never going to get ulcers. And so the question became why? Why is it this organism causes ulcers in some people and in severe cases can lead to stomach cancer, but in many other people, the organism is not a problem? And so scientists speculated that maybe in the past that most humans had this organism living inside us, and it was maybe a beneficial relationship, right? That we had this organism, right, that we lived symbiotically together. It was part of our normal flora. And at some point, because of dietary changes and lifestyle changes, now this organism is causing problems in some people. So we really don't know for sure, but many ulcers are caused by this organism and is treatable with antibiotics. Azospirillum is just an environmental organism. It's just there to mention it because it's spiral-shaped. Aerobic rods and cocci that are gram-negative. So when it comes to gram-negative cocci, these do tend to be pathogens, which is one of the reasons why we never use a gram-negative coccus in lab, because they're pathogens. And we'll talk about some of these later, but some gram-negative rods, Pseudomonas and Legionella. Legionella we've talked about, a respiratory pathogen. Pseudomonas, we did use a species of Pseudomonas in the temperature lab. We use Pseudomonas fluorescens, which is basically harmless and does produce a fluorescent pigment. But the Pseudomonas you most often hear about as a pathogen is Pseudomonas aeruginosa. And so this organism, it can cause infections like ear infections, swimmer's ear is often caused by Pseudomonas, other skin infections. But some people, it's a small percentage, maybe 5% of people, you can find Pseudomonas in their throats. So when you do a throat culture, you can find Pseudomonas. And I actually had a student once who had Pseudomonas in her throat. And I had to go look that up because I'd never seen such a thing. But it is found in a very small percentage of people. Apparently, it causes no harm in them, but it is a pathogen. It's one that we used to use in lab, but it is pathogenic. But we used to use it because it produces a green extracellular pigment. So Pseudomonas aeruginosa produces a pigment that is secreted into the environment. So it was a good example of an extracellular pigment. And Pseudomonas aeruginosa actually has a very pleasant, fruity smell. So it actually does smell good for a bacterium. And we will see and talk about that on Wednesday's lab when we look at growth patterns in lab. When it comes to Neisseria, we'll later talk about an example of Neisseria gonorrhea, which causes gonorrhea. Rhizobium and Agrobacterium are environmental organisms associated with plants. We'll see that Rhizobium is actually beneficial to plants that are infected with it, and Agrobacterium causes tumors in plants. But we'll talk about how scientists have been able to exploit the ability of Agrobacterium to penetrate plant cell walls. But we'll get to that in a few minutes. When it comes to the facultatively anaerobic rods, these are the gram-negative rods. We have a separate part of this lecture devoted to them, so we will get to them later because they're very important. They're pathogens as well as part of our normal flora. Rickettsias and chlamydias. These are gram-negative bacteria. What is special about them is that they are small and they're obligate intracellular parasites. So that means they actually get inside of cells to cause an infection. Almost all the other bacterial pathogens are extracellular, so they're on the outside of your cells and tissues. But Rickettsias and chlamydias actually get inside. Now, when it comes to Rickettsia, when I ask students, what disease does Rickettsia cause? There's something the students always say. And what disease do students always say? Rickets, which is a nutritional deficiency disease, right? It's not a bacterial pathogen. So an example of a rickettsial disease is Rickettsia rickettsii, which causes Rocky Mountain spotted fever, which is transmitted by ticks. When it comes to the chlamydias, we will talk in a few moments about chlamydia specifically, but chlamydia and the now new and closely related genus Chlamydophila are human pathogens, and we will talk about chlamydia as a sexually transmitted infection, but it's also a common cause of blindness, bacterial blindness. And so we'll talk about that shortly. Mycoplasmas, you already know about mycoplasma. By default, they're gram-negative because they have no cell wall. There's no peptidoglycan to hold on to the crystal violet. And since there's no cell wall, there's no definite shape, so they're pleomorphic. And Mycoplasma pneumoniae is probably the most common cause of bacterial pneumonia in the United States. When it comes to the gram-positive cocci, staph, and strep, we have a separate part of this lecture on them, so we'll wait until we get there. Endospore-forming rods and cocci. So yes, there are some gram-positive cocci that make spores. It's not common, it's not typical. Remember, most of the spore formers come from the genus Bacillus and Clostridium. But there are some cocci like Sporosarsina, which is a coccus that makes spores, but that's not common. So we'll talk about, well, we've already mentioned Bacillus multiple times because we've seen two species of Bacillus, Bacillus cereus and Bacillus subtilis. And remember, soil has many different types of Bacillus in it. When it comes to Clostridium, it is also found in soil. And remember, Clostridium is a gram-positive rod that makes spores. And we'll talk about three species, tetani, perfringens, and botulinum. So Clostridium tetani causes tetanus. I'm sure you've all heard of tetanus. You know, what's the sort of classical example of how you get infected with tetanus?
From like a rusty nail, like stepping on a rusty
A
nail.
rusty nail. Right, stepping on a rusty nail. That's always the example that's used. And so it's not that the nail is rusty. You know, rust is just iron oxide. It's that the nail's been exposed to the environment. It's probably down in the soil. Remember, Clostridium makes spores. And so the spores can be injected into you if you step on the nail. So, but there's a big difference between stepping on the nail and just scratching yourself with it. Scratching yourself is not a big concern. But stepping on it and getting a puncture wound, that is a concern. And so when it comes to Clostridium, last week we learned something else about it, which would help explain the difference between a dangerous puncture wound versus a harmless scratch. So what did we learn last week about Clostridium? What did we learn last week? Yes, it does produce CO2. And what else about it, Bryant? What did you learn about its oxygen requirement? What did we learn about the oxygen requirement for Clostridium? It's an obligate anaerobe, right, Bryant? So an obligate anaerobe. So the difference between a deep puncture wound and a scratch is that the scratch is aerobic. And so there's no concern about the spores germinating. But a puncture wound delivers the spores deep into your tissues, which is actually anaerobic. And so the spores will germinate and start an infection. And so the classical symptoms are sustained muscular contraction. That's what tetany refers to, sustained muscular contraction. And so if your jaw muscles are contracted, you can't eat, right? You can't chew, you can't swallow, it's hard to drink. And so you would die from starvation and dehydration if you got infected with tetanus. Of course, today they could keep you alive with IVs until you recover. But we do get vaccinated against tetanus, right? You've all gotten tetanus shots. which you're supposed to get like every 10 years or so because the immunity doesn't last. But whenever you get a tetanus shot, they're not immunizing you against the bacteria. You're being immunized against the toxin produced by the bacteria. It's the toxin that causes the sustained muscular contraction. And so your immune system is perfectly capable of eliminating those bacteria. But as we'll learn much later when we talk about the immune system, it takes 7 to 10 days to mount an immune response. You'll be dead by then. So we vaccinate against the toxin so that you can resist the toxin and give your immune system time to kill the bacteria. So it's an unusual kind of vaccine where we're vaccinating against the toxin because it is the toxins produced by the bacterial pathogens that do the damage and cause the symptoms of a disease. When it comes to Clostridium perfringens, this is associated with very septic wounds where the tissue starts to die, and then these bacteria will grow because dead tissue is anaerobic. And so this causes gangrene. So gangrene. And the skin often bubbles up because CO2 gas is produced, and it causes the skin to bubble up. So we often call this gas gangrene. So, but it does happen when tissue begins to die and becomes anaerobic. But Clostridium botulinum, that's the very, very interesting one. So a Clostridium botulinum, which causes botulism, you may have heard of botulism. So Clostridium botulinum produces a very deadly neurotoxin. And the classic example had to do with improperly canned foods. So whether they were being canned at home or, early canning processes, the factories didn't always do the best job. If you didn't cook your food at least 30 minutes, the spores could survive. And then in that sealed can, it was anaerobic, so the spores would germinate and the bacteria would grow and produce the botulinum toxin. And so, if you saw a can that was bulging, the tops of cans are normally concave, but if it was bulging out, you should never open that or eat that or anything because it's highly contaminated and most likely with botulism. And so some people would still use it and just cook it, thinking they could cook it to make it safe. But the botulism toxin is a pretty tough toxin. You really have to boil it a long time to destroy it. And so people would eat the toxin. It would paralyze the diaphragm so they could not breathe. And so they would basically suffocate and die. Several years ago, a strain of Clostridium botulinum was discovered in a patient, and that strain produced the deadliest version of the botulism toxin known. In fact, it was so deadly that the scientists did not release their findings until they had an antidote, because the concern was that this toxin was so deadly, even in very tiny amounts, that they were worried about terrorism. If terrorists got a hold of this, they could poison a water supply and kill, hundreds of thousands of people. So they were afraid to release their findings until they had an antidote. But the botulism toxin is one of the deadliest toxins known to humans. And so only humans would do this with a deadly toxin. Humans inject it into their faces because this is Botox, the botulism toxin. And they inject it into their faces to paralyze the muscles because it's the muscles that cause wrinkles, right, when you smile and make facial expressions. And so people use a deadly toxin for the sake of vanity, right, to look younger. Now, there are some actual medical uses for Botox. If you have a muscle that's impinging on a nerve and causing pain, you can paralyze that muscle for a while, and that will alleviate the pain. They use Botox for severe migraine headaches, again, because some of the muscles on your skull are very tight, and that tension causes pain and leads to migraines, and so they can use Botox to help alleviate certain migraines. So there are actual medical uses. But whoever had the idea, let's stick it in someone's face and see what happens. Fortunately, the toxin does not migrate. It stays where you inject it. Otherwise, all those people would be dead by now. But leave it to humans to use an extraordinarily deadly toxin, for vanity, right? So they don't wrinkle. When it comes to some other gram-positive rods, these are non-sporing rods. So there are some gram-positive rods that do not make spores. An example, Lactobacillus acidophilus. So Lactobacillus is a gram-positive rod, but it does not make spores. And the species acidophilus is actually one of the bacteria that you add to milk to spoil it in a good way to make things like yogurt. But the genus Lactobacillus is also an extremely important part of the normal flora of women. And we'll talk about this later, but the vagina is heavily colonized with Lactobacillus, which helps to protect the vagina. We'll talk more about that later on. And then we have some other gram-positive rods called irregular non-sporing rods. These are things like Corynebacterium, which starts out life as a rod, but as it gets older, it tends to get rounder, look more like a coccus, so it's pleomorphic. And Corynebacterium diphtheriae causes diphtheria, which is something we do vaccinate against. So not all gram-positive rods make spores, but when it comes to spores, most of the bacteria that make spores are gram-positive rods. Mycobacteria, we've talked about this a lot. They're not technically gram-positive, although they stain purple with the gram stain. But remember, they don't really have quite enough peptidoglycan to meet the criterion for being truly gram-positive. They have those thick waxy cell walls, the mycolic acids. They do tend to grow slowly, and it is harder for antimicrobial drugs to penetrate that thick waxy cell wall. We mentioned Mycobacterium tuberculosis. I'll talk more about that later in the course. Also, Mycobacterium leprae, which causes leprosy, and the harmless form we use in lab, flea. Now, there are some more unusual bacteria out there, some that have special structures, appendages, like Caulobacter. And Caulobacter produces this stalk, an attachment structure, technically called a prostica. And they actually hook together and form a rosette. So kind of like a flower, shape when you look at it in the microscope. So there are some more unusual kinds of bacteria out there. When it comes to the archaea, remember, they're not in the domain bacteria, but I am mentioning them here because they have different kinds of cell walls. These are unusual organisms. They tend to love and need extreme environments like heat and salt. There are some bacteria that photosynthesize, but most of the bacteria that photosynthesize do not make oxygen. They're just making energy for themselves, but no oxygen. The one group of prokaryotes that do make oxygen are the blue-green algae, also known as cyanobacteria. And so, they really are more kind of like algae, but when they examined them microscopically, they noticed that they were missing something, a nucleus. And so by definition, the cyanobacteria must be prokaryotic. Even though they're big, you can easily see them. They form large mats on stagnant bodies of water. So they're very large, but technically they're prokaryotic. And they also make oxygen, like plants. So here we're revisiting agrobacterium. And so this is a tumor. It's called crown gall. And you may have seen these at the base of some trees. Every once in a while, you'll see this large growth coming out of the base of a tree. Well, that's a tumor, and the tree has been infected with this organism. So plants also do get cancer, they get tumors. But when scientists discovered what was causing the tumors, they found this bacterium. And what fascinated the scientists was the idea that this bacterium can penetrate the plant cell wall. And so once scientists realized that, they learned how they could use agrobacterium to carry genes into the plant cells and genetically modify plants that way. So scientists found a way to use a natural plant pathogen as a tool for biotechnology. When it comes to Azotobacter and Rhizobium, these bacteria have the ability to absorb atmospheric nitrogen and convert it into compounds like nitrates. Nitrates are fertilizer for plants. Azotobacter are free-living bacteria that live in the soil, and so they absorb the nitrogen, they fix it, and they make things like nitrates which fertilize the plants. Rhizobium is a symbiotic bacterium that lives in the roots of plants. And here we see these root nodules, which are swollen areas that are filled with Rhizobium bacteria. And what the Rhizobium bacteria do is they absorb nitrogen from the air and convert it into compounds like nitrates, which fertilize the plants. So that allows the plants to live in soils that are very poor in nitrogen. And the typical plants which have rhizobium are legumes like beans, peas, and peanuts. And so they can grow in very nitrogen-poor soils. Neisseria, here we see these gram-negative cocci. Neisseria meningitidis causes a form of bacterial meningitis. And in general, bacterial meningitis is worse than viral meningitis. And then we have Neisseria gonorrhea, which causes gonorrhea. And so again, a gram-negative coccus, they do tend to be pathogens, so we never work with gram-negative cocci in lab. The Vibrios, I just want to mention because Vibrio cholerae causes cholera. And so these bacteria are found in water. And whenever there's a natural disaster like a hurricane or typhoon, often there is a cholera outbreak in the developing world. And so these are curved or comma-shaped rods, but they do cause cholera. And don't worry about parahemolyticus. Campylobacter, once again, see this organism, it does have one polar flagellum. And what do we call that arrangement? One polar flagellum. So it's on one end. And what's that arrangement called? Monotrichous, correct. So monotrichous. And the fancy word for food poisoning is gastroenteritis. Helicobacter has a cluster of flagella on one end. And what do we call that arrangement? What do we call a cluster? Ophotrichous. And so, as I mentioned, Helicobacter pylori is a common cause of ulcers, and in severe cases, it can lead to stomach cancer. So, gram-negative rods. So, the family of enterics. So, the enteric bacteria. So, these are all facultative anaerobes. Remember, that means they love oxygen but do not need it. Many of them are very important medical pathogens, but they are also part of our normal flora. So we do find these organisms living in the intestines of humans and many other animals. They can be motile or non-motile, And even this can be variable within the same species. So not every member of the species is either motile or non-motile. Many of the enterics produce chemicals called bacteriocins, which are proteins that kill other related species of bacteria. So many of these bacteria are trying to kill off the competition. And so they produce these bacteriocins as a way to kill their competitors. So there's a lot of competition in the bacterial world. Many of these enterics produce toxins. A toxin that is secreted is an exotoxin. And so it's the toxins that do produce the symptoms of a disease. And when it comes to identifying your gram-negative rods, lactose fermentation is a common characteristic that is used because certain of these gram-negative rods will ferment lactose and some others do not. So we can use that as a characteristic to help identify them. So here we have several examples of gram-negative rods. Enterobacter, which is a normal flora microorganism. It is responsible for many UTIs. And what's a UTI?
Urinary tract infection.
Right, a urinary tract infection. And so a very common cause. Escherichia, in particular E. coli, is part of our normal flora, but as we've already learned, there are some nasty strains of E. coli, like 015787, which produces a potent enterotoxin. And enterotoxin is an exotoxin that acts on the intestines. Klebsiella pneumoniae can cause pneumonia, but you really have to aspirate it into the lungs, so that's not easy to do. And Klebsiella is one of those organisms, it's very, very slimy, and it's a small oval rod. So it's an oval rod, easily confused with a coccus. Proteus, small, very motile gram-negative rod, also associated with urinary tract infections and wound infections. Salmonella, remember, the species have been reclassified. Most of them are enterica, right? So Salmonella enterica, and then serovar or variety, whatever else, Typhimurium. And so Salmonella causes food poisoning or salmonellosis. It can also cause a severe form of food poisoning called typhoid fever. Serratia, or more technically, is pronounced serratia because in Latin there is no sh. So there's no sh sound in Latin, but most people just say serratia, but technically it's serratia. And this is the common cause of nosocomial urinary tract infections and respiratory tract infections. What does nosocomial mean? What's a nosocomial infection? Nosocomial. Where do you get it? From the hospital. So these are hospital-acquired infections. Shigella produces a toxin, the Shiga toxin, which causes severe diarrhea. And so the Shiga toxin gene has been shared. And the organism that received it was the E. coli 015787. It's got the Shiga toxin gene in it, and that's what makes that strain so nasty. And then Yersinia pestis is an organism with tremendous historical impact. It caused the bubonic plague, which killed countless tens of millions of people in Europe and Asia and the Middle East. It's transmitted by fleas. And people today still do occasionally get the plague. It's just treatable with antibiotics. So those were the gram-negative rods. just sort of interesting organism here, Delovibrio, is an organism, a bacterium that preys on other bacteria. And we see here the Delovibrio is actually attacking and penetrating into this other bacterial cell. And when it does that, it gets inside and basically digests the inside of that bacterial cell. It reproduces, makes more of its own, cells, and then kills that host bacterium. So there's a lot of competition in the bacterial world. So there's other bacteria that basically eat other bacteria. Again, just to mention that there are some photosynthetic bacteria. Most of them do not produce oxygen, so they're anoxygenic. Don't worry about any of this stuff here. The blue-green, Clostridium. Again, we know a lot about Clostridium, right? Gram-positive rods, spores, obligate anaerobes. And the spores they produce are terminal spores. So they're at the ends of the cell. So they're terminal spores. This is not the traditional staining technique. There's no malachite green here because, as I mentioned, malachite green fades away over time, so it's no longer used commercially. So the gram-positive cocci, staph and strep, extremely important. They are medically important because they're very pathogenic. They are common nosocomial infections, so you get them from the hospitals. They are both becoming extremely antibiotic resistant. And when it comes to Staph aureus, in particular, there's a type of staph that's extremely dangerous. Anybody know what that dangerous kind of staph is? What's that dangerous staph? Anybody know the dangerous staph? Ever heard of MRSA? MRSA, which stands for methicillin resistant Staph aureus, or more commonly multiple resistant Staph aureus because it is resistant to many, many different kinds of antibiotics. It's becoming more and more of a problem because it's becoming more common and it's very difficult to treat. Very antibiotic resistant. Now, Staph aureus, it can also cause things like food poisoning. And in fact, Staph aureus is a common cause of food poisoning. If you ever got sick several hours after eating something and you had severe diarrhea for 24 hours and then it went away, that was most likely staphylococcal food poisoning because it's fast and furious. It hits you, you have severe diarrhea, and then in about 24 hours, you're over it. Salmonella food poisoning to last several days or longer. Streptococcus, same concern, a gram-positive rod, nosocomial infections, more and more antibiotic resistance. But Streptococcus can cause a variety of diseases like the common strep throat, scarlet fever, pneumonia, and the flesh-eating strep is real. There are certain streptococci that produce toxins which dissolve away your flesh. One of these toxins is beta-hemolysin, and as the name tells you, hemolysin, it destroys red blood cells. So if you had a blood infection with streptococcus, you're in serious trouble because it's destroying your red blood cells. Mycoplasma, remember, no cell wall, they're pleomorphic, and they're also very small. 0.1 to 0.24 micrometers is very, very small. And Mycoplasma pneumoniae, a common cause of bacterial pneumonia. When it comes to chlamydia, and now the closely related Chlamydophila genus, Chlamydia trachomatis, it is the most common sexually transmitted infection in women in the United States. It's very common. but it's also the #1 cause of infectious blindness in the world. And what happens is if your eyes get infected, it causes your upper eyelid to turn inward and your eyelashes scratch up the cornea. And so there's no way to repair that damage. And so you go blind. A Chlamydophila pneumoniae causes pneumonia, and so does Chlamydophila psittaci, which is transmitted by birds to humans and can cause pneumonia. But remember, these are tiny intracellular parasites. Borrelia and Treponema, two important spirochetes that are pathogens. Borrelia causes Lyme disease, which is transmitted by ticks, and Treponema pallidum causes syphilis. And here we see a beautiful electron micrograph of a cross-section of one of these spirochetes. And so you can see all the axial filaments in cross-section. And remember, you have the cell body and the sheath. And the axial filaments are attached to one end of the cell, but they're free leaving on the other. And that causes the bacteria to kind of spin like a corkscrew. The cyanobacteria, the blue-green algae, remember these are prokaryotes that photosynthesize and make oxygen. And they're very large. They are motile, but they use a different type of motility called a gliding motility. There's a genus called Oscillatoria, and the long, slender green cells actually slowly move back and forth. They oscillate. But remember, these things are big, easy to see. There's just no nucleus. When it comes to size of organisms, remember that we typically use micrometers, but there are some extraordinarily tiny bacteria and some extraordinarily large bacteria. An extraordinarily tiny one are the nanobacteria, 0.02 micrometers. Extremely small. They're basically just a chromosome with a bit of cytoplasm and a few ribosomes surrounded by a cell wall. Very, very tiny. But we do find nanobacteria in nature in rocks. We also find them in people. Thiomargarita is on the opposite end of the scale. It's huge, 750 micrometers. That's 0.75 millimeters, or 3/4 of a millimeter. You can see these with the naked eye. And so how they work is that they store sulfur. So in the center, they have a giant sulfur granule, and the cell membrane and cytoplasm is spread very thinly all around that gigantic granule of sulfur. Epilopicium, another enormous bacterial cell. It's at least 0.5 millimeter long, so you can see it with the naked eye. It's bigger than paramecia, which are eukaryotic protozoans. And in fact, when scientists first saw this in the gut of a surgeonfish, they just assumed it was a protozoan because it's so huge. But when they looked more closely, they realized there's no nucleus, there's no Golgi, there's no endoplasmic reticulum. It's a prokaryote, huge for a prokaryote. So there are some enormous prokaryotes, some really, really tiny ones, but those are the extremes. Now, remember when we talked about the environmental plate in that first week of lab? And we did samples from the desktop, your fingers, teeth and gums, soil, water, the air. And you saw a bunch of stuff growing on those agar plates. What you saw growing at most represented 10% of all the stuff you put on those plates. The problem with all the rest of the microorganisms is that they will not grow on plain, TSA. They need, special nutrients to grow. So how do we know that most of what we put on the plates actually did not grow. Well, if you perform PCR and you look for things like ribosomal RNA genes, what you find is that there's actually a lot of different microorganisms present. It's just that most of them will not grow on standard types of nutrient media. But we know they're there. It's just that we don't know how to grow them. So there is a tremendous number of microorganisms that are always present, far more than can actually be grown, but they are there. When it comes to the archaea, I do want to mention them because they're really amazing microorganisms. They often love or need extreme environments like the hyperthermophiles, which can thrive in boiling water and also boiling toxic water full of sulfur. Examples, pyrodictyum, so pyro referring to the heat, and sulfolobus referring to the sulfur. Methanogens are not really extreme, but they are obligate anaerobes that produce methane gas. And then the halophiles, right? They need that extremely high salt concentration, up to 30% sodium chloride. And has anybody heard of deep sea thermal vents? Deep sea thermal vents? Anybody heard of those? Nope. So these are ecosystems deep beneath the oceans, and they're near what are called black smokers, these volcanically active structures which spew out hot, toxic water. And the toxic water has a lot of sulfur in it. And what they discovered were these amazing communities of organisms that live down there. Tube worms, like what we see here, that are 5 feet long. I mean, all kinds of, unusual crabs and shrimp and fish. And this ecosystem, no light reaches it. There's absolutely no photosynthesis whatsoever down there. It's pitch black, extremely high pressure, also very hot and toxic. And yet there's all this life that exists down there. And these giant tube worms, if you notice, they're red, and they're red because of these microorganisms that live in their tissues. And it's the microorganisms that extract the sulfur from the water and use it for chemosynthesis to make energy. So these whole communities are chemosynthetic, not photosynthetic. So no light, no photosynthesis. But you have these amazing life forms down there. So incredibly inhospitable conditions. And yet there's all these amazing forms of life. So that gives us hope of finding life on other planets. Because the only absolute requirement for life is liquid water. It must be liquid. It can be boiling hot. It can be freezing cold as long as it's still liquid. And so, because we know that life forms can exist under incredibly harsh conditions, it's possible that there is life on other planets as long as there is some liquid water. No sun is needed. So, any questions or comments? If you don't know about deep sea thermal vents, make sure you look them up and see, because they're really amazing, communities of life.
How do people get down there to get like a
They had special submersibles, equipment which can travel down there. And when they were sending one down to look around, it saw these things and all these life forms, and it really stunned scientists when they discovered these things down there, because they did not expect to find anything. But there's these amazing communities. Yeah, it's really amazing. So later on, take some time to look up the deep sea thermal vents and just see these amazing forms of life that are down there in pitch blackness and incredible, incredible pressure. In fact, it's not possible to bring up those life forms to the surface because they all deteriorate, because they need that intense pressure. That's how they evolved. And you try and bring them up, and they all fall apart because not enough pressure. So it's kind of hard to study them. So any questions on all these different kinds of microorganisms?